Thank you, thank you very much, Kai. And I suppose I equally would like to echo, will say my sincere thanks for the invitation to speak at this really interesting event, and also, to compliment the organisers in, putting this, the bull, essentially a very unappreciated animal, will say, front and centre on the agenda. And I think we need to see more and more, I suppose, reference, will say, to bulls, both from a natural service and AI perspective, in cattle fertility symposium in the future. So just in terms of what I'm going to speak to you about, it's really, I suppose, a synopsis of a lot of the work we've been doing in recent times, looking at factors affecting, I suppose, sexual development, we'll say, and subsequent fertility in young bulls. Now, a lot of the work we've conducted, I suppose, has a focus on dairy-bred animals and potentially animals maybe destined for AI. But I think, the vast majority of the concepts are equally applicable, and certainly the scientific background to it is equally applicable to beef animals and bulls in natural service. So in terms of background, I'm going to just give, I suppose, as a brief introduction as to why we might be interested in trying to advance sexual development in the bull calf. And I know that you've had talks yesterday, we'll say, on, I suppose, management of replacement heifers. And again, it's an area we're working on ourselves too. And also then just a quick recap, I suppose, on the main aspects of postnatal sexual development in the bull calf leading up to puberty. And in terms of, I suppose, the main focus of the talk will be essentially early life nutritional management on endocrine or hormonal and molecular responses within the hypothalamic pituitary testicular axis, which is essentially the brain testes axis, we'll say, that really governs and dictates, we'll say, sexual development and subsequent fertility. We'll look at the effect of nutrition on age of onset of puberty and the overall trajectory of sexual maturation. Also, some work we've done in looking at post-pubertal semen characteristics as a consequence of earlier life management. We'll also very briefly cover some very recent work we did in I suppose, combination with partners at INRA in France on the, I suppose, methylation patterns within sperm. And then also, we'll say, in terms of some particularly in vitro based measures on bull fertility. And if I have time, I'll just briefly outline some additional work we conducted recently on looking at nutritional supplements and specifically fatty acids and bull fertility. So then, just taking, I suppose, a slide from a review there from Mississippi State University, where they just, I suppose, looked at some of the main factors affecting bull fertility. And I suppose they can be broken down into inherent, we'll say, intrinsic factors such as genetics. Obviously, we'll say breed, and there's significant variation within breed, and it's puberty and sexual development, and also fertility is, heritable. It's also subject into environmental conditions, whether they be weather, whether they be disease exposure, or indeed nutrition, which would be one of the main ones. And I suppose really the combination of those two then can also lead to epigenetic effects, where essentially the environment, will say, that the bull or the animal is exposed to can modify the functionality of its DNA. And these authors, will say, recommended back a number of years ago that, in looking at this, we should really take, I suppose, a interactive systems, I suppose, biology type approach, because, these are very complex traits that we're dealing with, lots and lots of different influences, both inherent and exterior to the animal, and we need to take a holistic view when we're trying to uncover maybe the underlying biology and make progress. And that's hopefully what we've tried to do ourselves. So then in terms of sexual development, just a couple of definitions, and more than likely Professor George Perry will develop some of these in his talk on bull breeding soundness evaluations later. But we're, essentially in most of the literature, we'll refer to almost an age-old definition there from Wolf from Pennsylvania State, going back to the 60s, who defined puberty as an ejaculate containing, greater than 50 million sperm with greater than 10% progressive linear motility. And sexual maturation has been more recently defined, we'll say, from John Kastek's group in Calgary, as an ejaculate with greater than 30% progressive motility and greater than 70% morphologically normal sperm. So when I refer, I suppose, to those two traits, those are the definitions that I'll be using. And again, there are studies that have shown or have looked at, would say, I suppose, the age at onset of puberty across different breeds. And, as we see with heifers, essentially the later maturing breeds typically are older at onset of maturity. Within the more British type breeds, would say, they would be, again, older, we'll say, maybe than some of the dairy breeds. But again, significant variation within breed as well. And also significant variation in terms of the actual weight of animals across breed and within breed at puberty. Well, one of the factors that's reasonably consistent across breed, we'll say, and within breed is the actual size of the scrotum or the scrotal circumference at the time of puberty, hovering in around, we'll say, 28, 29 centimeters, we'll say, or about 10.5, 11 inches in diameter. And I will refer to that again in the context of our own work later on. So again, some work from John Castelic's group in Calgary, where they looked at, I suppose, within Charolais and Angus and crossbred bulls, I suppose, the spread in age of puberty and also then subsequent sexual maturation. And again, you can see a variation there over 3-4 weeks, we'll say, there between when the animals started to become pubertal and when all animals were more or less pubertal. And based on the prior definition, sexual maturation, again, a longer spread, but averaged roughly about 50 days, those beef-bred animals, would say, after puberty, that they were, I suppose, defined as sexually mature. So why are we interested in events in sexual development? Well, we all know, we'll say, that particularly in cattle, generation interval limits genetic gain. And, in its simplest terms, the generation interval can be defined as the average age of the parents when the offspring are born. And essentially, I suppose, despite, the adoption of genomic selection, which has particular advantages maybe for some of the low heritability traits such as fertility, I suppose the power of that technology can only be really realized where the gametes are available, we'll say, and where animals are available for breeding. So again, it's important, we'll say, that both puberty and, I suppose, subsequent sexual maturation, we'll say, are brought forward as early as possible so that these genetically elite animals that can be identified through technology such as genomic selection, can be used as early as possible and ideally in their first breeding season. And that obviously, we'll say, is, much more important in seasonal production systems like we have both for beef and dairy over here in Ireland and in Northern Europe. But, if you take the beef system, it's ubiquitous in terms of it's a seasonal system. So there's a significant cost, like there is in rearing the heifer, in rearing these young bulls. And again, we don't want to be waiting, we'll say, excessively long before we can get those animals into production. And we do also know then that semen, I suppose, production from these peripubertal bulls, that they have lower semen quantity and quality compared to their more adult contemporaries. Indeed, they only produce about a third to maybe a half of the semen yield of a mature bull in their first year as a stud, for example. And again, we also know, we'll say, that the maximum, I suppose, daily sperm production per unit of testis weight in the first 20 weeks or 5 months or so post-pubertal is only a fraction of that. So they don't reach it until they're about 5 months after puberty. So essentially, that has implications as well for young bulls in natural service and the cow to bull ratio that we can afford to use. This is just a bit of work that we did in conjunction with colleagues Sean Fair and Pat Lonigan, where we looked at data from an AI bull breeding centre and looked at the effect of bull age across a number of different parameters. So essentially, as you might expect, as the animals got older, up to about 4 years and over in this study, the volume of semen, we'll say, produced per ejaculate increased. But when you looked at concentration, it kind of, I suppose, leveled off after about the first year. But obviously, when you combine the two of these together, the amount of sperm available in any one ejaculate, we'll say, obviously increases, we'll say, with bull age up to certainly about 4 years of age. And on top of that, the quality of that sperm, would say, when it's used within an AI context, both pre-freeze and post-freeze, would say, also increases up to about 3 to 4 years of age. So again, I suppose highlighting the lower quality, essentially, would say, of semen from younger bulls. and the importance of trying to bring forward puberty and sexual maturation so that we get, adequate quality, we'll say, in the first season. So this is just I suppose, a schematic for any of you, that are following the literature in terms of bull fertility. You'll be well aware of it. Essentially from Norma Rawlings from the University of Saskatchewan in Canada, where they look at the trajectory of, I suppose, sexual development in the bull from birth right through to onset of puberty. And again, you can see it can be divided into, I suppose, 4 different phases, really. Infantile phase in the first couple of months where there's not a lot happening, into a significant proliferative phase there where you get major proliferation of the 2 main cellular types, the Leydig cells, which produce testosterone, and the Sertoli cells, which are key cells that actually nourish the sperm and ultimately dictate the spermatogenic, potential of a bull. In other words, the amount of sperm that bull can produce. And they proliferate dramatically, we'll say, after about two to probably 6-7 months, we'll say, of age. And essentially, a lot of that is driven during that period by an early rise in the gonadotrophin luteinizing hormone produced in the anterior pituitary, the brain, under the influence of GnRH, or gonadotrophin releasing hormone, from the hypothalamus. And when I speak about the hypothalamic pituitary testicular axis, that's what I'm really referring to. And it's thought that the actual magnitude and the timing of this LH rise, this early LH rise, has significant consequences for, I suppose, the timing subsequent, we'll say, ensuing of actual puberty. So obviously, we'll say LH is driving testosterone, and then testosterone is having a negative feedback again on these 2 gonadotrophins. So we know from some of our own work and other work in the literature that early life nutrition can influence sexual development. And we know equally, we'll say, and you heard of this yesterday, we'll say, in terms of its effects in the heifer as well. And we do know, we'll say, from, that early life or improved nutrition can advance sexual development, we'll say, and puberty onset. It's mediated through complex neuroendocrine signaling, I suppose mainly really, we'll say, from hormones produced in peripheral tissues like the liver, like, for example, we'll say fat tissue, signaling to the brain, and the brain in turn signaling to the testes, and starting that cascade of sexual development. However, the timing of nutritional intervention that apparently dictates the potency of in response in the heifer isn't quite maybe as clear, or hasn't been in the past anyway, we'll say, for bulls. So with that in mind, we reviewed the literature and we looked at the few studies, we'll say, that had looked at that, and overall, across both beef and dairy studies, there was evidence to say that the better the plain nutrition, the better the metabolic status of the young calf, particularly in the early months of life, the quicker they became pubertal, and obviously were producing semen of adequate quality. But yet there was still, I suppose, some ambiguity there as to the exact timing, or the optimum timing, and also the optimum, I suppose, level of performance. So we conducted a number of studies, we'll say, and 2 of those were published a couple of years ago in the Journal of Dairy Science, to look at the effect of plain nutrition during the pre and post 6 months of age phases. So again, during that kind of infantile, we'll say, and also that proliferative phase, we'll say, that I talked about earlier, in Holstein Friesian bulls on age of puberty and post-pubertal semen production. So again, we divided about 80 bulls into at 2 weeks of age into a high plain nutrition, which was essentially very high milk replacer allocation per day, plus ad libitum access to concentrates. These are all delivered using an electronic feeding system. And a more lower or moderate, maybe would be a better term, would say, plain nutrition, which would be more standard practice, for bull calves, where they were fed about 500 grams of milk powder per day and about access to about a kilogram of concentrate. So after 6 months of age, they were subdivided within that high and low group into their continue on a high plain nutrition, which was essentially ad libitum access to high quality grass and high quality and good quality concentrate feed, or they were maintained, or they were swapped over, essentially, we'll say, to the opposite treatment. And the low here being, good quality grass again, but only about a half a kilogram of concentrate per day. So again, as you might expect, we got significant divergence in terms of growth rate. And again, really, even though you can't see it clearly here, but from about 6 weeks of age onwards, there was statistically significant divergence. At the end of, I suppose, the first 6 months of life, there was about a 60 kilogram difference between the highs and the low. So again, as I said, after 6 months, we divided them, we'll say, then into the 4 groups, and the highs kept growing, we'll say, in terms of a linear growth phase. The lows equally, we'll say, growing at a much lower rate of gain, and the other 2 crossover treatments, we'll say, somewhere in between. So at the end of that, I suppose, 40-week period, more or less, there was about 140 kilograms in terms of live weight. And again, this would be the period roughly, we'll say, at about 40, 45 weeks of age that you'd expect puberty to be occurring. We also measure scrotal circumference during that period, and again, it more or less mapped, we'll say, I suppose, the divergence in growth rate, with the animals on the high plane nutrition having a larger scrotal circumference than those comrades on the low plane nutrition, and obviously the other 2 groups, would say, in between after 6 months of age. So scrotal temperature is often, I suppose, a topic of debate, we'll say, when it comes to high planes nutrition, we'll say, for these young bulls. And obviously we know, we'll say, from all the very good work that the likes of John Kastelic has done, we'll say, at the University of Calgary, we'll say, over the years, and studied, I suppose, the influence of temperature, we'll say, on spermatogenesis in particular in great detail. We know that the test days, ideally, we'll say, the temperature should be about 2 to 6 degrees lower than the core body temperature. And we also know that increased testicular temperature, irrespective of the cause, can cause a depression in semen quality and can lead to either, I suppose, subacute, we'll say, or acute infertility. And the duration, we'll say, and the decrease in semen quality seems to be related to the severity and duration in terms of sperm morphology. And it takes about 6 weeks then to recover normal spermatogenesis, and the resumption of fertility after that can indeed take longer. And we also know, we'll say, that I suppose increased scrotal temperatures can be the consequence of a number of different issues. We'll say either contraction of disease, we'll say, where there's inflammation, injury, or indeed, we'll say, increased fatness of the scrotum. So with that in mind, we're interested in looking at the impact of our own treatments, we'll say, on, because again, as you've shown already, there was significant divergence there in terms of growth rate, we'll say, and performance of the animals, from about 16 weeks of age right through to about 40 weeks of age. And as you can see, overall, the animals, I suppose, on the higher plane, particularly early on in the first, 3 or 4 months, the animals on the high plane nutrition had higher or a greater scrotal skin thickness, which would suggest greater fat, I suppose, development, we'll say, within the scrotum. But all treatments began to converge in around, we'll say, 36 to 40 weeks of age. So again, when you look at the differences here, you're only talking about less than half a centimeter in scrotal skin thickness. And again, it's indicative of the kind of low, I suppose, levels of body fat in general, we'll say, in these type of young animals. So essentially, I don't believe that greater insulation of the testes as a consequence of high planes nutrition is having any potential major effect on spermatogenesis. Scrotal surface temperature then was measured, we'll say, using infrared technology. And again, we looked at the gradient, which is essentially the change in, I suppose, temperature from the pampiniform plexus or the cone, we'll say, the vascular cone here at the top of the scrotum, the neck of the scrotum, right down here, we'll say, to the caudal epididymal area, essentially where the sperm are being stored. And you can see there, overall, there was no interaction, we'll say. We looked at it over 3 months, we'll say 3 consecutive months, when the animals were between about 28, we'll say, and 36 weeks of age. And plane nutrition did have an effect in that it was slightly greater gradient, we'll say, for the animals on the high plane nutrition. And I suppose the overall average temperature, we'll say, was just slightly higher, we'll say, for those animals. But again, you're talking about a fraction of a degree Celsius, we'll say, in those animals on the higher plane nutrition. When we looked at the effective month, again, from an Irish perspective, we never get excessively high temperatures. But if we do get them, I suppose June and July typically, we'll say, is when they occur. So again, month had very, very minor effects. So this is just, I suppose, a diagrammatic representation that I took from a review paper that we wrote there a couple of years ago, looking at the various, I suppose, endocrine or metabolic, we'll say, interaction that occurs between signals coming from peripheral tissues, such as I suppose, the stomach, we'll say, the liver, and the pancreas in terms of insulin and fat tissue, and the hormones they produce and how they might signal either directly to the testes or indeed, we'll say, to that blood-brain axis. Which ultimately, as I said already, dictates, I suppose, the timing of sexual development, the speed of it, and also has I suppose, implications for subsequent fertility. And we looked at some of the key metabolic hormones in terms of how they might have diverged as a consequence of the different nutritional regimens that we subjected our calves to. And as you can imagine, some of the key ones like IGF or insulin-like growth factor 1 was much higher in those animals on the high plane nutrition than those their comrades on the lower plane nutrition. Equally, insulin, obviously, we'll say, which its main function is to regulate blood glucose, equally was much higher, we'll say. And again, both those on their own right have been shown to have positive effects, we'll say, on aspects of sperm production and in terms of testicular development. We looked at two what they call adipokines, or I suppose hormones produced by fat. We found, we'll say, much, I suppose, less conclusive evidence. And again, this would be indicative of, I suppose, again, as I mentioned earlier, the low amounts of fat, we'll say, in these young animals. So leptin has been positively associated in the past with earlier sexual development, and adiponectin has been inversely related to earlier sexual development. In other words, it inhibits it. But again, no great evidence, we'll say, of divergence there at that stage. And that would be consistent with a lot of studies in calves, where up to about, at least 6-7 months of age, it's hard to see any effect of nutrition on leptin, at least using the assays that we have available. We looked at the reproductive hormones, and again, this is over on a weekly basis that we measure them. On this particular graph, we also looked at it much more, I suppose, tighter intervals. FSH overall would say no effect, clear effect on nutrition, and it declined over time, as you might expect. LH, we didn't see in this particular study, would say, a clear, I suppose, increase in LH, but in a sister study that we conducted at the same time, where we measured it in a bit more detail, we'll say, we found, I suppose, a mirror of that, I suppose, schematic that I showed earlier there, that at about 10 to 14 weeks of age, there was a clear increase in testicular or in LH production from the anterior pituitary, which again, as I say, drives testosterone from the testes. And as you can see here, in those calves on the high plane nutrition, from about 20 weeks of age onwards, there was a clear increase, we'll say, and dramatic increase in the amount of testosterone that they were producing, which is again indicative of testicular development overall, and obviously would say positively related, would say, to the onset of puberty. So with that in mind, and getting down to the meat of the matter in terms of, what do these different diets, would say, both pre and post 6 months of age, how do they affect, would say, sexual maturation? Well, I divided this. It's a table, and apologies if the quality isn't maybe that great. It's lifted directly from the publication. Well, I've divided it, would say, into the two columns on the left-hand side here are those animals that are on a high plane nutrition for the first 6 months of life, and those two columns on the right here of the red line are those animals that are on a low plane nutrition during the first 6 months of life. Okay, so again, if we look, we'll say, overall age of puberty, the animals that were on a high plane nutrition for the first 6 months of life were pubertal on average about 30 days earlier, we'll say about a month earlier, and a range from about a month, we'll say, to 6 weeks, depending on which treatments you compare, than the equivalent animals that were on a low plane nutrition during the first 6 months of life, even those that were swapped over at 6 months to a high plane nutrition. So I suppose if you have an animal that's on a low plane nutrition or not, I suppose, meeting its nutritional requirements in the first 6 months of life or the first month, early life development, feeding that animal more or offering it more feed, we'll say, after that is not going to bring forward. So the race or bring forward, I suppose, puberty or sexual maturation to any great extent. So essentially the race is ran after about 5 to 6 months of age in terms of dictating age of puberty. In terms of weight of puberty then across the treatments, there was significant variation. They're up to 140 kilograms between the animals on the low plane nutrition when they reach puberty versus those on the high. However, as I mentioned earlier there, the one thing that was quite consistent was the actual scrotal circumference of the animals at puberty across those very divergent treatments. And again, it hovered in around about an average of about 28, 29 centimeters, which was quite conservative. And, so it's obviously a trait, we'll say, that's quite consistent, we'll say. And obviously, why it's used, so much, we'll say, within the normal bull breeding soundness evaluations. Age of sexual maturation occurred roughly on average about 30 days after puberty was defined. Then what we did, the next thing we did was that we slaughtered later in the experiment, we slaughtered those animals at about between 17 and 18 months of age. And we weighed the test days, and again we seen a residual, even though they're all from puberty onwards managed exactly the same and fed, a fairly high concentrate diet. There was still a residual effect there on testicular weight, even 6 months more or less after puberty had occurred, in that those animals on the high plane nutrition as calves will say had larger test days than their contemporaries on the lower plane nutrition. We looked at epididymal weight, and again remember that's where the epididymis is the part of the testes where sperm are actually stored. There was no effect of early life nutrition treatment. We looked at Sertoli cell density, and again, as I mentioned earlier, the Sertoli cells are really key cells in terms of dictating the spermatogenic potential of a bull. So essentially, potentially the amount of sperm will say that the bull can produce, and they're apparently set at about 6 months of age. So essentially, after that, they're not really subject, we'll say, to kind of environmental influences to any great degree. So we see no difference, we'll say, across the various treatment groups in terms of Sertoli's volume density, or indeed, we'll say, the proportion of different stages of the more mature sperm. So I suppose in summary there, what the data is telling us is that, we can advance puberty consistently, we'll say, by at least a month, we'll say, by preferential treatment of the calf. In a calf that's, I suppose, light for its age at 6 months of age, feeding it more, no matter even giving it ad lib access to meal, to concentrates, is not going to advance puberty even a day, essentially. But beyond that, then, there doesn't seem to be any major effects in terms of, I suppose, testicular development, we'll say, or indeed potential spermatogenesis. So another interesting hormone that we looked at there in conjunction with colleagues at the University of Nottingham in the UK was insulin-like protein 3. Or peptide 3, I should say, which apparently it's a peptide hormone that uniquely reflects Leydig cell function. And again, Leydig cell function, as I said already, will say the Leydig cells are key cells within the testes that, dictate, we'll say, testosterone production in particular. And we found, we'll say there, that in our calves on the high plane nutrition, that those had much higher circulating concentrations of this particular hormone than those on the low. And equally, there was a negative at both 4 and 8 months of age. There was a negative relationship, we'll say, there between the concentration of this hormone, we'll say, and the age of that animals, will say, determine puberty. So again, it might be something that might be worth, I suppose, looking at, or in terms of, I suppose, biomarkers for animals with the propensity to actually have earlier puberty, earlier sexual maturation, and ultimately be used maybe in their first breeding season. So going back to this, I suppose, old schematic again, again, as I say, so we have now shown that preferential treatment from a nutritional point of view during this key period here, up to about 6 months of age, does advance puberty. So the next question then really was how is this manifested? So to get a greater insight, we'll say, into the potential, we'll say, effects of prevailing plane nutrition during this early calfhood period, we have conducted a number of studies looking at kind of, I suppose, in depth, the kind of molecular biology or the underlying biology, we'll say, that might be dictating this across a number of different tissues. So to date, we've looked at calves, feeding calves, divergent planes of nutrition up to about 12 weeks of age, collecting tissues at that stage, and again, prolonging it to 18 weeks of age. So again, within that, I suppose, key window between about 10 and 20 weeks of age, that is apparently the key phase to where the sexual machinery or plastic, more or less, would say, are responsive to prevailing nutrition. And we collected different parts of the key parts of the brain that kind of interpret or receive, messages from hormones, we'll say, from those peripheral metabolic tissues, as I said already, and also, we'll say, other metabolic tissues like fat, for example. Apologies. We've subjected those tissues to in-depth kind of molecular biological analysis. And again, some of this work is still ongoing, but I'll briefly, we'll say, outline some of the key aspects of it. So we looked at fat. I mentioned earlier there that we didn't see big differences in leptin, which is, I suppose, a protein hormone produced by fat. But when we looked at the actual fat cells that would be producing that leptin, we've seen major differences between our high and low planes of nutrition, in terms of both the number of cells and also the size of those cells, even at 18 weeks of age. So those calves on the high plane nutrition had much greater fat cells and also greater cell diameter. So again, in theory, they should be producing more of those, I suppose, positive protein hormones that might influence, we'll say, early sexual development. We also then looked at the test days, we'll say, and subjected it to in-depth histology. Again, calves on the high plane nutrition had almost double the weight at test days at 18 weeks of age. Seminiferous tubule diameter, again, the seminiferous tubules is really where the sperm are developing, we'll say, again, was greater, we'll say, in the calves on the high plane nutrition. When we looked at some of the very early sperm cells, there was more of those at 18 weeks of age in the animals on the low plane nutrition than on the high plane nutrition. But when we looked at the more advanced stages of spermatogenesis or sperm cells, in other words, closer to, I suppose, a sperm capable of fertilizing an egg, there was much greater proportion of those, we'll say, on the high versus the low plane nutrition calves. Interesting, we'll say, enough, even though we didn't see it when we collected the test days at 17, 18 months of age, but at this young age, the Sertoli cells, which I keep going back to again, really important in terms of dictating the spermatogenic potential, the amount of sperm that a bull can produce potentially in its lifetime. There was evidence at 18 weeks of age that those calves on the high plane nutrition had a greater number of those cells than their contemporaries on a lower plane nutrition. And equally, I suppose the density of those cells was also higher in the calves on the high plane nutrition at this young age. And again, looking at the lumen scores, which is again, I suppose, a parameter, we'll say, or a variable used to look at the development of these seminiferous tubules at different stages. The higher score here being the, I suppose, the greater level of maturity. And we found that those calves on the high plane nutrition, dictated here with the blue bar, had higher scores for, I suppose, greater, lumen development, we'll say, than their contemporaries on the lower plane nutrition. We also subjected, as I said, the tissues, the testes and the brain tissues to in-depth RNA-seq. So essentially looking at the genes, we'll say, and how these genes are transcribed and they're produced, we'll say, ultimately proteins and hormones and the like, their functional capacity, essentially. And we found, we'll say, that in the testes, there was about 1,300, we'll say, genes that were different between, in terms of abundance, we'll say, or expression between their the high and the low planes of nutrition calves. And ultimately, most of those were involved in cholesterol biosynthesis. And again, cholesterol is a precursor for testosterone and androgen biosynthesis. So again, the machinery, we'll say, was upregulated or ramped up, we'll say, in the testes of those young calves that were on a high plane nutrition compared to the animals that were more retarded in development on the lower plane nutrition. We didn't see the same level of divergence in the brain, but we felt it was an earlier phase or a late phase of development. We were looking more downstream here at what was happening in the testes. We also looked in, we'll say, at 12 weeks of age, and we found that at that age, it was actually really the brain where most of the action was happening, and there was less action happening, we'll say, within the testes in terms of gene expression. So again, it looks like, we'll say, that I suppose the differential starts really much earlier on, maybe around 10, 12 weeks of age, and ultimately, we'll say, results in greater testicular development from about maybe 15 to 20 weeks of age, which really ramps up in terms of testosterone production and testicular maturity and spermatogenesis thereafter. We're also using, I suppose, fairly in-depth statistical tools, more or less, to try and make sense of all this biology and ultimately identify what are the kind of key genes, we'll say, that are dictating, this earlier puberty, and can we then ultimately, within the context of genomic selection that I mentioned earlier, use these genes and variants within the genes then to maybe identify animals that are predisposed to earlier puberty, and ultimately that you have a better shot at utilizing those animals in their first season, whether it be for AI or for indeed natural service. Some of that work has already, and this was published in the Professional Animal Scientist there a couple of years ago, identified a number of genes when we kind of, I suppose, throw everything into the melting pot and look across a load of tissues, we'll say, with our outcome variable here being Sertoli cell development. And as I said again, key cells that dictate the number of sperm, we'll say, that a bull could potentially produce. And we found a number of kind of key genes that were that seem to dictate or influence, we'll say, Sertoli cell development. And a lot of these, we'll say, have been shown in other studies to actually be associated with bull fertility as well. So which is always encouraging. So I suppose our objective is to follow that in more detail. this is just something I threw in. We'll say it's a bit of work Pat Lonigan and myself have done. We'll say, as I said already, we're equally looking at the similar treatments in heifers, and we published a study there, and I was glad to see it actually cited in one of the talks yesterday. There, Alan Kelly was the first author looking at some of our work. We'll say it was published in Journal of Animal Science there, literally in the last month or so, in heifers, but as part of that work, we fed heifers differentially. There are higher low plane nutrition, something similar to the bull work that I talked about earlier, from 4 to 8 months of age. And, you know, and again, working off some of the very good work that was done at Texas A&M in particular, we'll say, in this area. And we looked at, we'll say, puberty thereafter then in those animals from 8 months, we'll say, right through to about 16 months of age. We bred those heifers then at 16 months of age after synchronization, and we slaughtered the calves. They were all bred to the same sire. We slaughtered them, we'll say, at about 100 days of gestation, recovered the male fetuses, and looked at a number of different parameters, including gene expression within the testes. So that's just a schematic there. It just shows, we'll say, I suppose, the percent pubertal across those diets, so the moderate diet versus the high plane nutrition. Again, as I say, this was all happening here prior to 8 months of age, and then they were all in the same plane of nutrition thereafter, and we monitored puberty right through to about 15, 16 months of age. And this is part of John Hazelin's thesis, and I'm glad to say George Perry was actually the external examiner on that PhD thesis. And essentially what it's telling us is that, you know, right through to about almost, we'll say, to the key time at about 15 months of age, when we would really need these heifers to be pubertal and submitted for breeding, you know, there's a clear advantage, we'll say, for the animals on the high plane nutrition in terms of the percentage pubertal versus their contemporaries on the low plane nutrition. So they didn't really converge right until the end, we'll say, of, you know, really after the age that we'd like to be submitting them for breeding. So I suppose we hypothesized in, you know, could there be, I suppose, a nutritional effect if we're recommending essentially better nutrition to these heifers? Does it have any effect at all on their progeny thereafter? So again, it was a kind of a look-see experiment. And as you can see there, we slaughtered these calves, recovered the fetuses about 100 days, and recovered the testes. And we looked at fetal crown rump length and also fetal weight. And again, no evidence, we'll say, I suppose, of the nutrition of the heifer. Their mothers will say at an early age, from 4 to 8 months of age, no kind of carryover effect, will say, on their male progeny, will say, at 100 days of gestation. However, when we again worked with some of our colleagues in INRAE in France and looked at the histology of the testes, there was a greater proportion of interstitial, we'll say, tissue in the fetuses from the low, the heifers that were fed a low plane nutrition between 4 and 8 months of age, and there was also greater seminiferous tubule density, which would be seen as a positive in terms of testicular development in the fetuses from the heifers on the high plane nutrition. And then when we went and looked at gene expression, you know, we found about 55 genes that were differentially expressed, and these were predicted to be involved in a whole, I suppose, host of different elements of the reproductive system across, we'll say, a number of key kind of areas that would be associated with, I suppose, gonadal development, we'll say, in the fetus. So that was just, you know, something interesting. Again, it's something that we need to look at in a lot more detail. So again, just moving on and looking at the effect of these early life nutrition diets, we'll say, on post-pubertal semen characteristics and fertility. So I suppose, again, the question, you know, hopefully I've shown, you know, that high plane nutrition does hasten the onset of puberty and sexual maturation. But does it have any effects then, we'll say, in terms of post-pubertal fertility or semen characteristics? And again, we looked at, we'll say, semen quality, we'll say, and volume related traits, quantity and quality related traits up to, from puberty right up to, we'll say, 17 months of age. And again, just showing here on the red, this just shows, we'll say, when, for context, when the animals on the high plane nutrition during the first 6 months of life reach puberty at about 40 months of age, or 40 weeks, sorry, of age, those animals on the lower plane nutrition were that bit later. So obviously you'd expect that, you know, in the weeks post-puberty that semen volume and quality or sperm quantity and quality should improve anyway. So that's just to put those in context. But overall, we didn't see any major effect, we'll say, certainly once both groups are pubertal, of of early life nutrition on either the volume or indeed the concentration of sperm being produced. And again, when we looked at, we'll say, when we looked at the interaction, again, there was obviously in the weeks post-pubertal, those animals on the high plane nutrition had greater quantity and quality, but thereafter then, they did converge. Sperm motility again was higher as a consequence of the earlier puberty in the animals on the high, but again after that, we'll say the two groups more or less converged again in terms of progressive linear motility. But when we put all the data together and we looked at, I suppose, the ejaculates that were collected from about 12 to 15 months of age, and we, I suppose, estimated the number of straws, had these bulls been in an AI center, we'll say, that we could have collected as a consequence of that earlier, I suppose, semen availability and quality from the animals on the high plane nutrition as calves in the first 6 months of life compared to their contemporaries on the low. We found that overall, we would have been able to collect about double the amount of straws, we'll say, from the animals on the high plane nutrition as calves, compared to those on the low. And obviously the commercial value, we'll say, then was about double, we'll say, as well. So from an AI perspective, that month, we'll say, or 6 weeks earlier, was significant, particularly if you want to get the genetics of that animal out, we'll say, in its first breeding season. And again, I suppose, go back to, from our perspective, on the dairy side in particular, we'll say it's a seasonal system. So again, if a bull misses his first, I suppose, spring, really he's not going to have an influence, we'll say, until the following year again, at which time he may well be passed out by a more genetically elite animal. So again, it has obviously, we'll say, positive connotations, but equally, I suppose, the breeding pressure that you could put a young bull under, maybe, we'll say, in his first season, we'll say, in the first number of weeks, when many of the cows, we'll say, will be submitted, we'll say, and bred even under natural service conditions. So then I mentioned already, we'll say, this concept of methylation, or I suppose it's a way, essentially a biological way, as to how the environment, which will include nutrition, might in some way affect how the DNA operates. So the DNA that the animal is born with, how it's actually expressed, we'll say, thereafter. So, I suppose the natural DNA, we'll say, that the animal has, or genetic merit, can be modified to some degree by the prevailing management of that animal, and particularly, as I say, nutrition. And again, we worked with colleagues at INRA in France to look at this using a particular, I suppose, technology, RRBS, or reduced representative bisulfite sequencing. This work was published there, in the last probably 2 months, we'll say, there. Again, we looked at ejaculates collected, we'll say, at 15 and 16 months of age, and also, we'll say, across the animals on the high versus the low plane nutrition as calves. And overall, we did see some differences. We'll say about 600 differentially expressed regions, we'll say, were identified, we'll say, within the sperm from the high and the calves on the high, or the bulls, I should say, that were on high or low plane nutrition as calves. No real effect, we'll say, of whether the ejaculate was collected at 15 or 16 months of age. There are a couple of those studies that show some effect of age, but maybe not quite as tight in terms of timeline as that. But we did see differences, really, we'll say, in some, I suppose, when we compare the high and low groups, in some genes that have been shown, to have clear functions in spermatogenesis, in other words, sperm production, so Sertoli cell function, and also the overall machinery, we'll say, that governs, we'll say, reproductive function. So again, it's something that we need to look at in a bit more detail as to how, nutrition might be actually affecting, we'll say, the DNA, we'll say, within the sperm, and equally we'll be interested in looking at within the testes and some of the other tissues as well. So it's again just adding another little bit of insight, we'll say, into how this, I suppose, early life nutrition might be affecting the trajectory of sexual development. So ultimately, we'll say, as I say, there was some evidence there, we'll say, that the methylation pattern of sperm can be affected. So then again, getting back, we'll say, to fertility, and I suppose this is a piece of work that we did at Pat Lonergan's lab at University College Dublin, and we looked again with, as I mentioned already, we had taken straws, we'll say, taken semen from those bulls, we'll say, when they were about 65 weeks of age or so, from the different 4 treatments that we looked at, and we subjected that to an IVF system. And again, overall, we got acceptable, I suppose, cleavage rates, acceptable, we'll say, early embryo development, but again, no evidence of a latent effect, essentially, of early life nutrition on subsequent fertility. So certainly there was no negatives, we'll say, from feeding those calves a very high plain nutrition right up to about 40 weeks of age, or a year almost of age. So from that perspective, that was, I suppose, encouraging, you know. So essentially, you were bringing forward puberty, but no negative effects, we'll say, on potential fertility, certainly within the context of an IVF system. And again, it's always reassuring to see when your data concurs with other published literature, and again, some data from John Kestelic's group, will say, that looked at something similar, and they equally found no latent or knock-on effects. So then, just briefly, if I have time, I'll just briefly mention some of the some work that we did looking at, I suppose, supplements, and particularly dietary polyunsaturated fatty acids, or lipids, or fats. Again, there's a lot of literature, will say, to suggest it across different species that feeding fat, will say, can have a positive effect in terms of aspects of fertility across, you know, different species, as say. However, you know, there's not much consistency, we'll say, across studies or indeed species as to how these, I suppose, factors, will say, are manifested. So we subjected bulls to 3 different treatments, either a control, no fat addition, a N6 based, we'll say, polyunsaturated fatty acid, safflower oil, or an N3 fish oil. So again, it would be the highest fish oil or omega-3 fed in the literature, we'll say, in terms of concentration. So we fed that, we'll say, there for a period of about 50 days to 50 bulls. They were all pubertal. They had all been tested, and they were all producing normal sperm, we'll say, at the start of the study. And we collected semen on an ongoing basis then up to about 17 months of age. And as you can see here, we'll say, across the different number of parameters that we measured, we'll say, both the volume, the concentration, and the motility of the sperm, and even both pre and post freezing, there was no difference or no evidence that these fat, I suppose, supplements had any positive effect. And we often get it from our producers here, we'll say, pedigree breeders, that, you know, are there kind of supplements, and fat is often put in there, you know, into the rations, we'll say, both for heifers and bulls. And, you know, we've done equally a lot of work down to the years looking at some of the similar type diets and heifers, and again, found no effect in terms of, you know, reproductive parameters or indeed embryo quality, we'll say, by feeding these fats in the diet, which, you know, obviously are expensive. So overall summary, Mr. Chairman, then, enhancing, I hope that I provided data to show that enhancing early calfhood nutrition leads to, I suppose, increased gonadotrophin secretion early in life, advanced steroidogenesis, in other words, testosterone production and testicular development, and overall an earlier onset of puberty and the availability potentially of saleable semen, or indeed a bull that can literally be turned out, we'll say, with cows. This is obviously mediated to a very complex, I suppose, interaction or interplay between metabolic signals coming from key tissues, we'll say, within the body, and specialized, we'll say, neuropeptides, we'll say, and hormones within the brain, and with knock-on effects, obviously, for the testes. We found an upregulation, we'll say, of different molecular pathways, we'll say, within the hypothalamus, the anterior pituitary at 12 weeks, and in the testes, particularly at 18 weeks of age. We also have some evidence for increased proliferation of Sertoli cells, certainly early in life within the testes, and potentially a latent effect, we'll say, of early life nutrition on the sperm, the DNA, we'll say, essentially, you know, collected, you know, almost a year later. There was no obvious effects, we'll say, and certainly no negative effects of improved plain nutrition on, I suppose, post-pubertal semen quality or indeed fertility. And we did see some positive effects, even though we're not sure exactly the biology behind it, we'll say, of actually feeding your replacement heifer better, you know, on potentially the sexual development of its fetus in utero, we'll say, later on, 8-9 months later. So again, it's something interesting, but, you know, some of these transgenerational effects that we hear about, we'll say, you know, are gaining more and more traction. Again, as I say, no particular effect or no evidence of a beneficial effect of producing or supplementing diets with polyunsaturated fatty acids in terms of any aspect of fertility we measured anyway. But ultimately, I hope that, you know, improved knowledge of these, you know, I suppose the underlying biology would say of how nutrition can affect puberty and sexual development can ultimately lead to better, I suppose, nutritional rearing regimens, both for natural service bulls and bulls destined for AI, and ultimately equally to identify biomarkers through identifying the key genes, would say that maybe nutrition is improving and that can advance puberty. So we can, I suppose, select for bulls that are more consistent in terms of going through puberty, sexual maturation earlier, and that are available for breeding at an earlier age. And again, trying to shorten that generation interval again. So we're also looking at, I suppose, a bit more in depth, would say, in terms of the underlying neuroendocrinology. We've also a large on-farm study looking at, I suppose, genes, different genes that are related to semen quality and puberty in young bulls. And we're hoping maybe to get some funding to look at in utero program, and again looking at the diet of the mother and its effect on the development of the ensuing offspring. So just to acknowledge the contribution of a lot of colleagues, would say, both funders, would say, as well as collaborators, particularly in University College Dublin and the University of Limerick, would say, in the southern part of Ireland. And I suppose with that, I thank you. And I suppose just to say that I suppose I don't just talk about bulls. I actually have a small pedigree Charlie herd, and very much enjoy, would say, working with those animals on a daily basis. So with that, I thank you very much for your attention and welcome any questions you might have. Thank you.
Thank you, David. Thank you for that very nice talk, very comprehensive and update on all the things that you and your colleagues are working on. So please go ahead and put your questions into the chat box. We're going to answer a few of these live, and then I'll ask Dr. Kinney to try to answer a few of them if he has time. So the first question is, should a beef producer that sells yearling bulls, can they creep feed those bulls as a mean of advancing puberty and the probability of that yearling bull passing a breeding soundness exam? So I guess, that question of whether does creep feeding kind of come in and play that high plane of nutrition and advanced puberty that you talked about?
Yeah, I think that's an excellent question. And again, it's something that we were hoping to get some funding to look again at the suckled calf, essentially. And it's obviously it's a subject close to my own heart. I think, based on our own work and based on work that John Kastelic would have done with beef bulls, albeit bull calves that might have been early weaned, but subjected to a high plane nutrition thereafter, concentrate based, there's no evidence that subjecting these young bulls, would say up to a year of age, would say to high planes of nutrition and resulting in growth rates up to about kilogram and a half or 1.6 kilos a day, that has any negative effect on their subsequent fertility. Now, one thing I will caution, and one thing that certainly we're very conscious of as well, is outside of what's supposed puberty and immediate fertility, other issues that might be, I suppose, a consequence of high planes of high concentrate type diets, high starch type diets, high potentially acetotic diets, that might potentially lead, we'll say, to either subacute and hopefully not acute acidosis, that those issues may well have, latent effects on the actual mobility, and the joint health and everything else of some of those bulls. So it's essentially affecting their longevity. And again, we don't have evidence to suggest that's the case, but it's certainly the feed management, of those animals is very, very important to make sure that we avoid, either subacute and certainly acute acidosis, because we do know, we'll say that those type of conditions, can have very negative effects, both for the locomotive ability of the bull, which is so important, obviously, we'll say, for natural service sires, but also, we'll say, on sperm production within the testes as well.
Excellent. Yeah, I think there's a lot to be done on the bull side of things that we still have a lot of questions still to ask there. But let me ask you another question that's come in. A lot of questions in regards to, we have a lot of people joining us from around the world today. A lot of questions about Bos taurus versus Bos indicus, beef versus dairy. Just wanted to know if you could just address that a little bit. And then a very specific question to that is, if we focus on beef breeds who are excellent at converting energy into fat, do you feel there's a maximum threshold for the plane of nutrition before and after puberty, with the idea that after a lot of those beef bulls reach puberty, they start to put down a lot of fat and semen quality kind of goes down as you look at them in long term in a sire collection facility. So a lot of different questions there kind of summarized into one.
Yeah, those are, again, very, very relevant questions, Kai. And like what I would say is, I suppose our data would, and again, if you look at the data coming from Canada, would say the same, that essentially in terms of bringing forward puberty, which is, I suppose, our main remit, would say here, and making sure that those bulls are available at the start of the breeding season, whether it be, for semen collection in an AI centre or indeed natural service, that there's no advantage in subjecting those cows to a high plain nutrition after 6 months of age. So essentially, if they're backward, for example, if they were suckling on a cow with a beef bred cow with little or no milk, we'll say, and they were light for their weight at 6 months of age, feeding them even ad lib creep feed, we'll say, thereafter is not going to bring them forward in any which way in terms of puberty. So, if we are supplementing those animals, we'll say, from a sexual development perspective, it's really the first few months of life that's key. Thereafter, obviously, we all, know that producers like to have bulls well grown, we'll say, for sale and the like, we'll say, and as I say, there's no direct evidence that feeding them at a sustainable level and achieving sustainable growth rates, we'll say up to about 1.5 kilograms a day, has any direct negative effects on their fertility specifically. But I think that, as I say, if we want to try to purely advanced sexual maturation, there's no advantage whatsoever in feeding them excessively beyond, we'll say, 6 months of age. And in terms of the actual breed differences, then I think our work was mainly done with dairy bred bulls. The work in Canada, we'll say, was done mainly with beef bred bulls, and again, extremely consistent, in terms of the results, and even in terms of the timelines and the kind of factors that we're seeing being affected by these higher planes of nutrition. And I expect, we'll say, Bos indicus versus Bos taurus, again, the underlying biology is very, very same. The actual age at which puberty might occur might be different, as it is in heifers, but overall, I think the principles will be the same.